in 2007, I was given a $3 million US fund, and I spent all that developing technology for immune repertoire sequencing. In the United States, we're also hosting a immunogenomics meeting in collaboration with Nature and Science magazine. And it's a very nice place. I also encourage you to join us. And your group founder actually was Felix Brandon visited us and gave a talk at the our immunogenomics meeting a couple of years ago. And we had Mark Davidson come to give a talk and international researchers. So I like to show these slides because this is my original thinking of why I started the immune repertoire sequencing. I thought immune system is the best doctor. They detect external threat and internal threats, and too aggressive with external threat, we have inflammation. Too weak, we get infected. Internally, too aggressive, we have autoimmune disease, and too weak, we get cancer. Two of these aggressiveness lead to tissue damage, and two of these weakness, something abnormal grows. And that's cover all the human diseases, either something broke or something growing. And doesn't matter what happened, they're supposed to be a group of disease-specific antigen expressed at a wrong time, wrong place, or wrong volume. And our immune system already made a diagnosis and tried to do something about it. So that was my original thought of why we should do this. I thought immune system has the best disease signal. Its specificity is great, the sensitivity is great, and the signal is in peripheral blood. So we started doing this. We published our first paper in 2009 on PNAS, but as an entrepreneur, I filed a patent in 2008. We licensed our technology to Sequenta in 2013, and 2 years later, Sequenta was acquired by Adaptome for $200 million. So we generate all kinds of data. So far, we have sequenced more than, 200,000 samples, and we have a lot of data in storage. But I like this picture the most. It was developed by Dr. Chunling Wang from Stanford, one of our team, and the key designer of all the pipelines and softwares. And each one of the little dots is a unique clone type or CDR3 sequence. And the size of these dots representing the frequency of that dot in a sample. And the left individual is normal and the right hand is a patient. So, but before I go further, I want to tell you there's two long-tailed distributions people not so familiar with. One is in one individual. We have done exclusive sequencing of one individual, taking sample from the same individual the same day, one in the morning, one in the afternoon, and the sequence 40 libraries, each library including 1 million cells, and you'll find 360,000 unique sequences. Of all the 40 sequencing run, we find 5 million unique sequences, but 2 million of that show up only once. Another 1.2 million show up only twice. Every time of the 40 sequencing run, you will find only 10,000 that consistently showed up. So in one individual, the distribution of a repertoire is long-tailed. Small percent show up higher frequency. Majority of that belong to the long tail. There's another long tail, which is in the population. If you sequence 1,000 individual, you will find 75 million unique sequences, and majority of that are private and not shared, but small percentage are shared at very high frequencies. So these are two long-tailed distributions showing you that immune repertoire is not a normal distribution. Therefore, many of the statistics that we developed to describing diversity, describing everything are not very fit for describing immune repertoires. So that is a warning sign. The long tail distribution also tell us the signal, the biological signal tends to be on the head, and the long tail, if you are not careful, it's a lot of biological or analytical noise. So the technical challenge is that you have to get inclusive enough, view the repertoire, otherwise you are seeing the long tail. You have to be unbiased, quantitative, and noise-free. And our approach is multiplex PCR, and I've been an entrepreneur and started 4 companies, sold 2, and most of all these companies are based on multiplex PCR technology. And we realize there are 4 difficulties. Each primer you design need to work. Each pair you design need to work with other pair. They don't amplify background, and they don't amplify themselves. And for each one of the difficulties, we develop technologies and patent these several generations of multiplex PCR technology, which I don't have time to get into. However, there's several review chapters that I wrote with Dr. Michael Lotz and describing the technologies as well as comparing different technologies and approach. And Dr. Lotz is also the one coined the words of Adaptome. It's a genome and omic, because we are not just sequencing a repertoire. A repertoire is T cell, B cell, one chain, all the CDR3 in a sample. Adaptome is all 7 chains of T cell, B cell receptors in a sample and the CDR3 distributions. So we have automated the process of a library prep with this machine that made by a sister company called iCubate, where you load your sample and put the sample in and make the library a few hours automatically. And one of the things we can do with this machine is to amplify all 7 chains of T cell, B cell receptor in one reaction from one sample. And that will generate 50 new data points. And each one of them could serve as a biomarker for disease associations. All 7 chain distribution, T cell, B cell ratio, alpha, beta, gamma, delta ratio, alpha, beta ratio. This is important because we find some individual alpha, beta cell, significant portion of the alpha, beta cell have a dual alpha, but no beta. We don't know what's the significance. The gamma delta subsets, the diversity index, essential index, which is I'll tell these in a few minutes, and B cell isotype frequencies, hypermutation status, and B cell interchain ratios. So all these data can be generated with one sample in one reaction automatically in a disposable cassette. So how do we use these clinically? Just like the economic field, there's a GDP, there's an unemployment rate, there's durable goods sales, there's a whole kind of indexes describing the economy. So we come up with some indexes to describe immune repertoire or Adaptome dynamically or statically. And the first 3 indexes can be used to evaluate health and general health without referencing to diseases. And sharing index is looking for disease-specific sharing. And in this case with COVID, and us and others did find specific signals. And the last 3 indexes are used for treatment guidance. So the first top 3 individual, you can see are healthy individual with nice distribution of a repertoire, both chronotype and essential index, which is measuring the public shared CDR3, the top 1,000 very public CDR3, how many do you have? It's a measure of the immune reserve. The middle line is acute diseases, chronic disease at the bottom, renal cancer on top, and the leukemia patients at the bottom. And so with this picture, I want to share with you that patients, especially have chronic disease, usually have these large clones. And we don't know what is antigen this clone specific for, but some of the patients, as you can see with the cancer, these clones are very stable and they don't change. And I want to remind people that our immune system is just like your bank account, have upper limits. Every time you fight a war, you've frozen up a piece of resources that it's not available for other things. So we need to be careful with your immune repertoire. So the wellness index is the accumulation of a diversity index, essential index, and chronotype index. And you can see from cord blood with aging, the wellness index decreases, and especially in stage 4 cancer patients. So the disease-specific sharing is interesting. We call it sharing index. Each one of these lines is a CDR3 sequence. Each column is a patient sample. The left half is breast cancer patients. The right half is control. So this is a study done very early, about 8 years ago, with Hong Kong, Dr. Eva Kong. And so we initiated a project called R10K, study patients and controls, identify the CDR3s, unique CDR3s, and see which CDR3 are shared predominantly among patients not in control. And then we get another validation sample to see how much is sharing with the signal already identified. So this is an example of TB. Each one of the red dot is a patient. Y-axis is a disease signal in that patient compared to a control group. This is a training data sets with a different data sets. We can still detect over 90%, 95% of the patients. Latent TB, half of them have signals. Other disease do not have signals. So the underlying hypothesis here are disease process expose some unique antigens, and population-wise, patients have the disease, have shared T-cell receptors. We did the same thing for type 1 diabetes, CMV infection, work with a French professor, and also IBD with UAB collaboration, liver cancer with Taiwan National University Hospital, breast cancer with Taiwan and Hong Kong collaboration, and pancreatic cancer with Johns Hopkins, heart disease as well. So the R10K project initiated about in 2012 is that anybody is open enroll, anybody have a sample can contact us, and we provide sequencing at Hassan Nafa, a nonprofit research institute, and the data is shared. The collaborator will be the first author to publish their information. And we have now so far 30-some disease under consideration and study. And that goes to the COVID. We published a paper with the Dr. Zhongnan Science Group in Guangzhou very early in February. We have access to 10 patients. We use Adaptome, that 7-chain analysis on peripheral blood, longitudinal samples to study these patients. And we identified some neutralizing antibodies from this group, which I'm going to talk about later. But recently, adaptive group working with Microsoft have studied more than 1,000 patients, COVID patients, and identified the disease-specific T-cell receptors. And actually, when we use our data to confirmed, and that signal was there and quite strong. So the treatment guidance, response index, we studied anti-PD-1 liver cancer patients, and responders and the progressors can be differentiated by this response index cutoff, say 20%. What this means is before the treatment, we got a patient's blood, and 4 weeks, 6 weeks, and 20 weeks after the treatment, we got a blood. And if you compare before treatment and 4 weeks after, if there's more than 20% of the top ranked clone types become upregulated, then you are most likely belong to responders. This is another study with pancreatic cancer, and this is a renal cancer. We have a tissue FFPE sample. Responders have more infiltrating cells than non-responders. And over time, responders also have more elastic, active turnover of repertoire compared to non-responders. They have a frozen repertoire that is not moving. And before treatment, we were able to identify patients have a better diversity in alpha chain, beta chain, and heavy chain for those responders. With chemotherapy of breast cancer patients, before and after the therapy, you can find B cell, there's no correlation before and after, and T cell, you still have a strong correlation, meaning chemotherapy in breast cancer patients heavily impacted the B cell, while T cell is relatively less impacted. Another interesting finding is this recent publication using peripheral blood before immunotherapy identified the responders and the non-responders by a diversity index. So COVID, in China, we mentioned we sequenced 10 patients and we published this paper. What we're showing that when you do 7 chain in the healthy control, this is a B cell heavy chain, kappa and lambda, and the immune repertoire of alpha, beta chain, gamma, delta chain. And compared to healthy patients have their T cell repertoire basically collapsed. And they do coming back later on. These patients' repertoire T cell coming back after a while. So our goal is to find neutralizing antibodies. But before we did COVID, last year, we have studied 7 individuals in our company with before and after flu shots. So before the flu shots, at day 0 and day 3, day 7, day 14 and 21, we measured the all 7 chain repertoire. And we can see around day 7, there's a strong B cell response. And their dominant clones also emerged around day 7, and their satellite clones. And these satellite clone will build a network because of the hypermutation and the class switch, and building this network around day 7. This is another individual, also very strong response around day 7. And in peripheral blood, up to 70% of the responders clone types are responding to this antigen. And around day 7, forming very strong networks. And this is my study. I also form a lot of network. That's my sample around day 7. So we learned day 7 is very important. How to looking for these antigen specific response is that there's a time window, there's a IgG, IgM class switch, and there are also hypermutation. And we developed the software ready to analyze. When COVID happened, we studied 10 patients, multiple time points. We also find around day 7, day 10, there's a strong B cell response. However, in flu shots, the B cell is more IgG, and in patients, it's more IgM and IgA with COVID. and we see very dominant response, and we see the network forming around day 7 as well. So after studying multiple patients, we pick these chronotypes that forming networks around day 7 and they have a class switch. And then we hand these clones to Dr. Darren Stanley in Japan. They have a software that based on known antibody antigen sequences can predict the docking distance between the candidate B cell and the viral antigen. So with this approach, I think finding a antibody from the repertoire is like finding a needle in the ocean. But by study 10 patients, we shrink that ocean down to a lake. By study flu vaccine, we learn how to looking for these. Then we shrink that lake down to a pan. And then after the software analysis, we have these needles in our hands and around 20 also antibodies. We did in vitro synthesis, we did ELISA analysis and neutralizing assays, and some of these antibodies can neutralize the various RBD domain. So that is happening in China. And at the same time, in April, we start to have patients in Alabama, and we are using single cell approach. We have developed 4 or 5 years ago that you can actually using flow cytometer to put single cell, and we use magnetic beads covered with a viral antigen, pull down the plasma cell from patients. We studied 30 patients over time, and we did a single cell analysis, find the heavy chain and light chain, did in vitro synthesis. This approach have been successfully used by NIH Patrick Duffy's group to study a malaria vaccine. And they gave us a tetramer pooled single cell, 500 of them. We were able to pair the heavy chain and light chain directly from the plate and did in vitro synthesis, expression, and that antibody combined to the antigen. So that is the approach down in Alabama. And in Europe, we just finished a manuscript with Dr. Chang Pen's group study a cohort patients in Italy and Netherlands and find out they are long-term immunity with these individuals. So that's wrapped up my talk. Hope I still have some time. The Adaptome Immune Technology Institute in Nanjing now have this instrument, can make libraries, and collaborating with people in China and worldwide to study disease signatures and for drug development as well. Thank you very much.
Thank you, Dr. Han, for a very nice talk. So if anybody online have a question, you can type your questions in the chatting room. So I will forward your question to Dr. Han. So before waiting, I think we still have some time. So I have the first question. So actually show us some data about identifying signatures of different diseases with your cohort, with your trained cohort. So I wondering that what's performance, such as specificity and sensitivity in your test cohort, if you have some, for those diseases, and which disease, according to your data, perform better than others? So that's my question. Thank you.
Good question. For all the diseases we studied, we can find signal. However, to validate that signal, we need a different cohort, and that's why we're expanding the study. What was your second question?
Which for disease status perform?
Oh, which disease? Actually, the infectious disease is more specific than autoimmune disease, for example, or cancer, because external antigen, you tend to find more specific signals.
Okay, thank you.
Such as COVID, actually, is actually the signal is quite strong, and you don't see sharing of with many people.
Okay, thank you.
You're welcome. I see Felix is online. Hi.
Yeah. Do you have any questions?
Well, yeah, hello.
Hi.
I was wondering, I mean, one of the problems I think, I'm not a physician or anything, but these autoimmune diseases, they're so complex, and you talk about multiple sclerosis, but there's 5 different, phenotypes there. Do you think that these repertoires could be used to differentiate the different subtypes, or people must be trying to do that a lot, I'm sure.
Yeah, I think we need a more sample to have a verdicts, but we, like I mentioned, the long tail distribution, the signal to be used are limited. If this TCR sequence is too frequent, it's useless, because if more than, say, 20% of the population has it, then everybody has it. So you must find the signal somewhat public, and then still disease specific. And so you are hoping their T cell repertoire recognize organ specific, tissue specific, internal antigen specific. And that require quite deep sequencing and also quantitative sequencing. One of the problem we find by studying all these years with immune repertoire is the noise is tremendous, and both biological noise and analytical noise. And so we need to figure out how to get rid of them. Unfortunately, recently we have using DAM PCR, a new multiplex PCR that allow us to label each one of the molecule with unique molecular identifier, and that get rid of lots of noise.
I sometimes wonder if it's also the metadata describing the disease phenotype needs to be better standardized, and
that's true. That's true.
That's not your job, but
I would love to join the efforts, and let's talk, and we can put the R10K data
I'd love to.
up to share.
Yeah, that'd be great. Definitely.
Thank you.
Yeah.
Nice to see you.
Okay. Thank you all for that.